start. I'm just going to let some more people in to the from the waiting room here. Perfect. So as you all know, we are hosting a research design series in February and March featuring weekly webinars on quantitative and qualitative research methods from guest speakers. Quantitative topics will be covered in February and qualitative methods in March. So our first session today is on quantitative methods from Dr. Holly Etchegary. So Dr. Etchegary is NL Support's academic patient engagement lead. She is also an associate professor of epidemiology at the Faculty of Medicine at Memorial. So her session is going to open up the webinar series to provide a brief introduction to some of the key clinical research designs, both observational and experimental. With that, I will hand it over to Dr. Etchegary to get started. So I'm going to stop sharing my screen, Holly.
Excellent, great. All right, well, thanks everybody. I'm delighted you could all be here. Let me just very quickly say that I certainly don't regard myself as any expert in all kinds of clinical research design, but we do teach a fair bit of this in our graduate program in clinical epidemiology, where I teach in faculty medicine. And so I hope I can say some things about it. And by the way, it's funny, when Julia and I were testing this out, I just realized that I have this, whatever's happening here, things are dropping in. So apologies if it takes me a couple of minutes to get from slide to slide. So yeah, so that is what we're going to do today. Have a think about, or a high level anyway, review, a little bit of a overview of some of the sort of common quantitative clinical research designs. And I thought we'd talk a little bit about some of the strengths and weaknesses of each, but importantly, try to set the stage for next week. So, or whenever our next webinar is, I forget if it's next week, sorry, or the week after, but Dr. Twells will be coming in to talk about research questions and why they're so important, and they really are, because all of us, when we're thinking about a research question, we then do need to think about the design. So hopefully you'll be able to link back to some of the things we talk about today. So do jump in if you have a question, and do feel free to type it in the chat. And between Julia and myself, one of us will see it for sure. So I'm going to start off here. So two very high level, two basic designs: experimental and non-experimental, really. In clinical research, when we talk about experimental studies, we're usually, not always, but usually referring to a randomized controlled trial or some sort of clinical trial. And the key feature of these, of course, is that treatments are assigned randomly. And you'll see that's really important. We'll come back to this later, but it is really important because that helps us ensure that our groups at the beginning are fairly comparable in their baseline characteristics, which matters, and you'll come to see why as we talk about that a bit more today. And Dr. John Fardy, who has long done clinical trials, long been a person who teaches in this program. He's a gastroenterologist with Eastern Health here, but he teaches in our ClinEpi program. He'll tell us all about clinical trials before this month is out. So today I'm just going to touch on them. But in general, when we're talking high level, experimental is one of the key designs in clinical research, and it's usually, as I say, clinical trial. The other type will be non-experimental, or what we call observational studies, and there are, a variety of different types of these. So cohort, case control, and cross-sectional, probably three of the most common we see, and those are the ones that we'll focus on mostly today. Or a case study, which you see sometimes. Sometimes you see a series of case studies, but in general, these are the non-experimental designs. So in these situations, as the name implies, non-experimental or observational, we as the investigator, we aren't really randomizing anyone anywhere. So we aren't controlling which people are in which group, so to speak. But you'll see what I mean as we talk about these in a little more detail. The prospective versus retrospective, I just want to make that point that oftentimes when we're doing certainly a trial, it's prospective, but the observational studies, The cohort studies are usually prospective. We're looking forward in time. Case control studies are often retrospective. We're looking back in time. So I just want to make that point. Some of them are looking forward, some are looking back. And that can introduce various advantages and disadvantages, right? And again, we'll talk about that in a bit more detail. Before I do that, I want to mention this to you, this evidence pyramid, and maybe most people are familiar with this, and I'll just have a graphic coming up on the next slide, which those of you who've heard of it will definitely be familiar with. It's a way of thinking about the sort of quality of the evidence and the amount of evidence available. And so what I mean by that is, I often get asked, or people will often say, well, which design is best? Get that question a lot. And the answer will always be, it depends, always. Every single time the answer is, it depends. It depends on your question. It depends what you're trying to do. It depends on the purpose of your research, right? So if you're trying to establish some cause or effect relationship, a trial might be best. But if that's not what you're trying to do, a trial won't be best, for example, right? So, but we think about when we teach, certainly within the medical school and teaching healthcare providers about research for the first time, particularly if they don't have a lot of background in it. This is a very common way of sort of teaching about the clinical quantitative research evidence, which is the better, if you will. So systematic reviews will be at the top of the pyramid. They're the highest level of evidence, and they're the least common. You go down the pyramid, the amount of evidence will increase. So we have more and more studies, but maybe not of quite the same quality. So you see here, these systematic reviews and meta-analyses so are at the top of the pyramid. I probably could have actually had like clinical practice guidelines on the very top, because they're often informed by these reviews of the literature and meta-analyses, which try to give us sort of a quantitative estimate of the effect of something. So if I say, well, is aspirin helpful to, reduce heart attack risk? A meta-analysis will give us some quantitative estimate of that, as opposed to saying, yeah, they're related, but it's a small effect. For example, we're not going to cover those today, but I just point out that most of the sort of clinical practice guidelines upon which a lot of treatment, decisions are made, and those clinical sort of support tools are based on the highest level of evidence. And we want them to be, don't we, when we're talking about life and death and treatment situations, right? So following on from those reviews and meta-analyses are the trials, the randomized trials. And the systematic reviews and meta-analyses are often of bunches of these randomized controlled trials. And you can see as we go down, then the cohort studies are the next level of evidence, followed by case control and so on. And those are the ones we'll talk most about today, the cohort and case control, a little bit about cross-sectional. So I just wanted to point this out to you. It's a very popular pyramid within evidence-based medicine, very well known to those who do that. Now, I wonder if you thought about anything else. I don't know if you did or not. I'm curious. So I asked the question. As you saw that pyramid, were you thinking, well, I don't see qualitative, evidence in there. I don't see anything about sort of lived experience as evidence, which is a new field now, by the way. And those of you who will know a bit about patient engagement or patient-oriented research, more and more we're seeing or having discussions about where lived experience and patient experience and family and caregiver experience falls, or when does it count as evidence, and how does that balance up against what we traditionally think of as evidence, which I've just outlined. What I will say, so I just want to make note of that in case you were looking at that and thinking, well, I don't see this other kind of evidence. I think that certainly for qualitative research, those systematic reviews and the meta-analyses, well, qualitative research certainly does have comparable reviews. So I forget the exact words, forgive me, sort of qualitative syntheses and meta-narratives of a body of qualitative research. So technically they could be included in those systematic sort of reviews. And I actually think that any of the studies in that evidence pyramid can be, and more today often are, patient-oriented or at least informed by patient engagement. So I'd like to hope that there's certainly some lived experience of patients in there. But I just want to make the point to you, that's our traditional sort of pyramid. I wonder if that will change as we go forward and we see more of this discussion about lived experience as evidence. So I just want to point that out to you. So any questions so far, everyone, before I jump in now to sort of talking about two of these observational types of studies, the cohort and case control. How are we doing so far? Making sense? I don't see any questions there in the chat, but I'll give you a couple of seconds in case there are any. I just realized how weird this sun is on my face, by the way. Sorry, I'm sitting in it's hard to complain though about sun on your face, isn't it?
I was going to say, we're happy about the sun.
Okay, so everyone feel free, type your questions in if something comes to mind. So let's jump into talking about these two. Let me check my time and see how I'm doing. These two types of observational or non-experimental studies. So cohort studies versus case control. So in a cohort study, participants are classified according to their exposure status. And that very much is the language of cohort studies. They're followed forward in time to figure out what the disease incidence is. Case control studies, on the other hand, participants are selected according to their condition. So they already have a condition or a disease. We're looking back in time to sort of look at the risk factors for that disease. Cohort, we're going forward. So people are healthy at the beginning. We want to look forward to see if they develop a disease. That's ultimately what we're saying. But you'll see this now in a bit more detail. So here, an investigator will define two or more groups of people, again, as I said, that are free of a disease or a condition, but differ somehow in terms of their exposure to a potential cause. So here's an example: women who use video display terminals and a second group who do not. You follow them forward in time to determine the incidence of negative pregnancy outcomes, such as stillbirths or miscarriages, etc. So you see how here are people who don't have a condition or a disease, but they do have a different exposure. So one uses these video display terminals, one group, and one group does not. But we have some hypothesis for whatever reason. I don't know if this is true, by the way, it's just hypothetical. But for whatever reason, if we have reason to believe that, you know, spending a lot of time on these terminals may impact pregnancy outcomes, then that's sort of our hypothesis. So we look for those two groups currently free of the outcome, which here is a negative pregnancy outcome. We follow them forward in time. A well-known example of this, by the way, if you want to see a really long-running cohort study, is this Framingham or Framingham. I always forget how it's pronounced, Frey or Fram. This has been a cohort study started in the US a long, long time ago, 1948. This study is still going, by the way. I didn't realize it was still going, but it is. Looking at the risk factors for cardiovascular disease. And when this started back in 1948, again, just like the previous example, you know, no one had heart disease in the beginning of the study, right? So the point is to move forward in time. In Canada right now, for example, we have the Canadian Partnership Against Cancer study, the Canadian Longitudinal Study on Aging. These are also cohort studies that are current, that are following cohorts of people forward in time and ultimately, and are measuring all kinds of things along the way, and will ultimately look at those outcomes of cancer, of cardiovascular disease, etc. I just wanted to mention this to you because whenever we teach about cohort studies, we have to talk about Framingham because it's famous. And this is a current link, actually. I was just in there myself last week. So interestingly enough, today, as we sit here doing this webinar, the people who took part in this in 1948, now their grandchildren are participants in this study. So it's pretty neat. So it's just a good example of a cohort study. And we've learned a lot, by the way, from that study. So I encourage you, if you have an interest in this, or if you need to study or do cohort studies, that's a good example. So this is the very traditional graphic that we use all the time in teaching about this. And for the life of me, I don't know where it comes from. Please forgive me. I cannot attribute the source. I've just been using it so long, I forget where it comes from. But you can see here at the beginning, right, you have these two groups. So here the example are smokers versus non-smokers. Follow them over time to compare outcomes. But notice, unlike the trial, we're not randomly assigning anyone anywhere. They simply are. They are a smoker or they are not, or they use a video display machine or they do not, etc. Right? Follow them over time. So let's talk a little bit about some of the advantages and the disadvantages of this. So, because part of choosing a design, I hope I don't go too far. Okay, there we go. Part of choosing a design really is thinking about, like, well, what are the good things about this? What are the strengths, and then what are some of the weaknesses? So you know how in a trial, for example, we might, we randomly assign a treatment. So let's say I have a brand new pain cream that you rub it on, your arm or wherever, and it magically, cures pain, okay? Or it's really good for lessening pain. So that would be a pretty good treatment, right? So in a trial, we randomize some people to receive it and some people to not. So a treatment, potentially a good treatment, is withheld from some people in a trial. Same thing. What about if my cream was actually really bad and had lots of side effects? Then I would be exposing somebody, right, to a potential hazard. So the cohort studies, you're not doing that. As the investigator, the team, you're not withholding treatment from anyone. You're not subjecting anyone to anything terrible. So those can be advantages in a cohort study. Did we have a question, or is someone just not muted? I'll pause in case there's a question. As I'm blinded by the sun. Okay, maybe just a muting thing. All right, so in a cohort study, those are advantages. The other things is you can, in particular, match participants for potential confounds. And we'll talk about that shortly in a little bit more detail about confounds. But what if, so let's say we were studying, as I gave the example earlier, about the women in the video display terminals and pregnancy sort of outcomes. So what if there are, and I'm sure there are many other things that influence pregnancy outcomes, right? I'm just looking at one particular thing, the use of the video display terminals. So what if by just fluke, because we're not randomly assigning people, I had people in the group who use the video display terminals, maybe they also have a bunch of other risk factors for bad or negative pregnancy outcomes. So then it'd be hard for me to know or to tease apart, if I observe the negative outcomes, what's leading to it. So that's part of what I mean by a confound, and how can we sort of match for them, which we try to do, right, as well. And Mike, I'll get to your comment there just one sec. Let me just finish off this slide here. We can technically be careful about our eligibility criteria, try to match as best we can, and we can use standardized outcome assessments. That's always a good thing to do as well. So for example, let's say in my pain cream example, I had a pain scale 1 to 10, it's well validated. These do exist in the literature, by the way. If I were to use that in both my groups, that would be better than having somebody say, observe someone and try to determine if they were in pain. It's a little bit more subjective, right? So you certainly can do that in a cohort study. The cohort studies also can be expensive because some of them can go for a long period of time. So, but they're generally less expensive than randomized controlled trials. They can also help you to estimate both the timing and the directionality of events, because some of these go on for many years, and even ones that go on for 3 or 4 or 5 or 6 years, which is comparatively much less than some of the bigger ones, can still help us estimate the timing of, A, X before Y and that sort of thing, right? So those are some of the advantages of the cohort study. And so, Mike, let me have a look at yours now. Validated questionnaires, yeah. Oh, yes. That's a very good question. Yeah, so Mike's question is about, so talking about study instruments. And as I was just talking about the pain scale, right? A validated sort of pain scale. So often we do try to use validated questionnaires, and we do that for a bunch of reasons. But one of them is we're trying to allow comparisons across studies and across provinces, countries, globally. So for example, when we talk about the systematic reviews and the meta-analyses, for example, as that highest level of evidence, sometimes we don't have enough evidence that use the same sort of survey or questionnaire to allow us to compile the evidence for, does my pain cream, have a lower lessening impact on pain? So one of the great advantages to using them is that they do allow comparisons across multiple populations. And sometimes there really is an advantage to doing that. So I think in terms of Mike, the questionnaires, and if patient partners have different suggestions that don't work well within the questionnaires, that is a conversation between the research team. No easy answer to that, by the way. Technically, statistically, methodologically, we are not supposed to change validated questionnaires, or again, that simply makes them not comparable. which is, not ideal often. The other thing about the validated questionnaires is that they allow us, they can be compared to population norms, which sometimes is really important. So for example, in the mental health field, we have a bunch of validated, post-traumatic stress disorder questionnaires, hopelessness, depression, anxiety, etc. And we know what the norms are in the general population. And these things have been rigorously designed so that we're able to say a score on that is above or below the general population, which means we may or may not have to intervene. So that's one of the other advantages. What I have done, Mike, in my own work, and it has worked well, so I throw this out as a suggestion. So, I think we do need to recognize that the validated questionnaires won't contain every single important content area that patient partners might think are important related to their condition. What I've often done in a case where I felt there was real advantage to being able to compare our results to others, so I wanted to use the questionnaire, I've included some form of open items at the end of the survey or in a separate one. I've allowed some way to ask the question that patient partners have suggested that content area is important, and given people a chance to share their thoughts in a qualitative manner. So that's what I've done to try to respect what they're saying and yet still retain the rigor and the comparison that I want for the data. I don't know that that's perfect. Last thing I'll say about it is this, because I think it is relevant to all types of studies. We now have a movement, not just in Canada, but globally, to develop, share, validate, talk about patient-reported outcome measures and patient-reported experience measures. So we do now have some validated surveys and questionnaires that really have been developed very much from what patient partners are saying and patients are saying. So I actually think that moving forward, we'll see, I think, a bit more of that in use. And actually our support unit, we're going to come back to this as well. I think, I don't know if we're going to do it this year or not, we'll see, but we've been thinking about that on our end as well to start to share some of those ideas. So I hope that that begins to throw out some ideas. Not a perfect answer. There's no easy, perfect answer to it all, but I hope that helps share some ideas in any event. Yeah.
Do you mind if I speak?
Yeah, sure, Austin.
Okay. When I registered for the event, there was a directed questionnaire I had to fill out. So is that the type of questionnaire you're referring to? Like a validated questionnaire, leaves, in my opinion anyway, even I was thinking about this when I registered for the event, I had to answer all these questions, and some of them are, I think some things are missing on some of the questions, but
okay.
I'm just saying, that's my opinion. Yep. Other people might go in there and look at it and say, which group do I belong to?
Right.
Listen, I've been told many times that I'm like a square peg that they're trying to fit into a round hole. I did not see that on the questionnaire, and I didn't feel comfortable putting it in the thing.
Yeah, yeah.
So I think that a patient's objective on even research questions is an important aspect.
Yeah.
Because believe me, I've spent my whole lifetime doing research, not in a medical way or not the way you're talking about, but in a different field altogether.
Yeah.
But anyway, that's just my opinion, and
yeah.
Thank you.
No, that's great.
Thank you for letting me speak up. Thank you.
Yeah, no, thanks, Austin. I appreciate it. I assume the questions that we're asking sort of before these webinars are kind of demographic type questions, trying to get a sense of, do we, who's our audience? Do we have a lot of students? Do we have a lot of patient partners, researchers, providers? Yeah. So I will just quickly say that those are generally not the type of validated questionnaires I'm referring to. The ones I'm referring to are generally very rigorously studied, conducted, multiple populations, and generally measure some condition or trait. So depression, social functioning, physical functioning. That's what I mean by the validated questionnaires. I'll just say whether or not, I see the other question there in the chat, are they ever rechecked to make sure they're still accurate? That's a really good question. And my quick answer to that is, I don't really know. I don't do the work of validating questionnaires. That is a study in and of itself. Let me tell you, that is a research project in and of itself. It takes a ton of work and a ton of data. But I do know that some of the really well validated ones, like the Beck Depression Inventory, the Hopelessness Scale, the SF-36, they've been around for a really long time and they continue to be used. So I feel like there is some mechanism for revisiting, but exactly what is it? I've got to say, I don't know. I've got to say, I don't know.
Yeah, I can offer a suggestion. I was involved with discussions with people out in British Columbia Institute of Health, and they've incorporated a reporting system out there for patients. So patients, when they experience a visit to an emergency or a visit to a doctor or whatever, they do have a way, patients have a way of responding to their experience in the healthcare system. But unfortunately, they, unfortunately or not, I don't know how they created the validated questionnaires, because patients are giving a reporting tool. They've developed a reporting tool for patients, but the patients are only allowed to report certain things.
Yeah.
You know?
Yeah.
So the option they give patients is, okay, go and discuss it in your own forum. To me, that's like telling a person to go and call open line and have a chat about all the. But anyway, I think I'll shut up for the time being.
No, but it's no, but listen, your point is well taken, and I will move on just so I don't go too far off. But I will say this: your point is well taken, and I think there is a movement now in Canada to better understand what's already out there in terms of these patient-reported experience measures, and to work with relevant groups of patients. Cancer oncology is probably the area where it's most well developed, I think. In terms of, well, what are the content areas that matter? I think we're going to get better at seeing those evolve over time. So that's a good another webinar for us. We should add that to our list, patient reported measures. I love it. Okay, let me get back to the cohort studies just so I don't go too far off. So one of the things about cohort studies, remember, we're talking about, you know, groups of people who are already identified, right? So people use the machines or they don't, people smoke or they don't. If a therapy or an exposure is popular, if most people have been exposed, it'll be hard, won't it, to find controls, people who haven't been. Confounding may be an issue. Have a look at this little diagram down here. So let's say, you know, I wanted to study this relationship between drinking coffee and heart disease, okay? So this is my predictor. This is the thing that I think is going to impact my outcome or my dependent variable, which is heart disease. By the way, I have no idea if coffee impacts the heart. I'm just making this up. But we do know, don't we, that cigarette smoking impacts heart disease. It's related, right? So imagine if I had my two groups of people, the people exposed to whatever it is I'm studying, here it's coffee, if I didn't somehow control for, well, do they smoke? Because smoking, we know, impacts my outcome, heart disease. So confounding in a cohort study can be an issue. You have to be really careful, think about it, and try to control for it. I know we don't have time to go into it all today, but I just want to flag it as a potential sort of disadvantage. If it's a rare disorder, you'll need a very large sample size. And I think cohort studies, by the way, can also be expensive. I know I've talked about trials, and indeed they are, but they can also be expensive, as in that one we just talked about, right? That's been going on for years and years. So those are the cohort studies. Let's talk a little bit now about case control studies. So these are the ones I was saying that are generally retrospective, often retrospective. And here, as opposed to starting out with two sort of healthy groups of people, if you will, you're actually starting with cases, people who do have a particular illness or condition. And then you have to choose controls, right? People who do not have that particular illness or condition. You're generally then looking at, well, what's the level of exposure between these groups and comparing them? Because obviously you think there's some exposure that's leading right to the illness. Again, this is the classic sort of diagram that, again, I forget where I got this, my apologies. So here, for example, patients with cancer, non-patients down here, You look back over time, taking histories, right? Looking at where they've been exposed or to what they've been exposed, drawing conclusions. So that's generally a little bit different than the cohort, right? Here we're looking back. And here people already have the outcome of interest, right? The illness of interest. So if we think about some examples of that, some of the early studies about smoking and lung cancer, this is really how we began to look at the link, because obviously we cannot, randomize someone to smoke, right? Or go to a tanning bed or spend hours in the sun. And so often we're looking back over time. So in that, the original studies about lung cancer were case control studies, looking back and comparing the history, for example, right? Often uses hospital records to look back over time. There's usually some form of sort of administrative data included in these case control studies. So when you think about it, I guess right then and there, you can see some disadvantages, right? Because if records are not fully complete, you may be missing some data, right? Because again, you're looking back over time. But the advantages of it, very good for studying rare conditions or diseases. And I think, if I'm not mistaken, I believe it was case control years and years ago, this design that looked at that relationship between thalidomide back in the 60s, thalidomide and birth defects. This was because think about it, it was a rare thing. And so when you have a rare disorder, rare condition, it's often the only design you can use, because a cohort study, you just wouldn't have enough people, right, to follow them forward in time, and it'd be too expensive. Often then less time is needed to conduct these sorts of studies, right, because the condition has already occurred. So it's generally fairly easy and cost effective. I actually don't think any research is what I would call easy, by the way, but generally speaking. And it lets you look simultaneously at multiple risk factors, which I do think is an advantage. So that's simple when we just had there up on the screen that time, the coffee drinking and the heart disease and then the smoking. Well, if you're looking back over time, you could look at anything, right? Smoking, education level, income level, a job, are they environmental exposures, and so on. So you can look at multiple things at the same time, which I do think is a good thing. So we often use these as an initial study that helps us establish an association or begin to suspect that there's some relationship, and that can be useful for moving forward with other research. But the main trouble with them is because you're looking back over time, often they're just not as reliable, right, as the studies that do record the data in real time. So think about it. One type of bias is recall bias, which is really makes sense, doesn't it? If a case control study is also talking to people, not just looking back over their hospital records, when you answer questions about your previous exposure to something, to a certain risk factor, I mean, we forget, right? We forget something that happened 10 years ago or 5 years ago, or even last year, in the case of some of us. The other type of bias that will happen is what we call selection bias. And so this is something we had to think about really in all research, to be honest with you. But you really got to think about the cases, the patients you're choosing, and the control group you're choosing. Do they really represent the condition under investigation? And we see this a lot when we have studies that are done out of teaching hospitals that are, affiliated with a med school, for example. That could happen here as well, because that really is a highly specialized setting. When you compare to the way the disease occurs out in the real world. Same with the controls. They also might not be typical of the population that you're trying to compare these patients to. I mean, people who are volunteering their data for this study may have a particularly high level of health motivation. So again, in these studies, and it's partly why they're not ranked quite as high as a control trial on the evidence pyramid, because we do need to think about these different sorts of biases, and it's hard to think about them all, sort of thing. But we will, generally speaking, begin when you're doing these studies, begin to think carefully about these things up front and ahead of time with the case control sort of studies. But again, can be very useful for certain types of things, particularly when you have rare sort of disorders and whatnot. Yeah, Mike and Austin, I see your comment. Yeah, I agree, Mike. I think the validation is catching up to patient-oriented research. I'm looking forward actually to learning a bit more about these validated patient-reported outcome measures, and experience measures, something I don't know a lot about specifically, but I'm looking forward to learning. And I think we're going to hear more and more. AI, we're going to hear that more and more as well, Austin. It's not my area at all, I have to say, but I do think that, so that could be a separate webinar for sure, but would have to be delivered by someone with some expertise in it, which would not be me. I think you're going to see it more and more. Where we're seeing it, I would argue now, is in a lot of the genetics and genomics type modeling studies. I hear about it a lot, but I have no idea how it's being used. But keep an eye open. I'm sure we're going to see more and more of that. Cross-sectional studies. This is probably the one I do most often. Most of my stuff is cross-sectional, done at one point in time. Often in the clinical research, we're looking at prevalence. So, we see these numbers released now all the time, right? How many have the COVID vaccine? How many don't? Prevalence of boosters in different groups, often done at one point in time. And most survey studies, I think, are also done at one point in time. Most. I know in the cohort studies, those big long ones that are happening over 10 and 20 and 30 years, they're doing surveys at different time points, but they're all cross-sectional, being done at one point in time, right? And that's the key definition there, one point in time. So there are definitely some advantages to this. I mean, first of all, it's easy, right? It's generally easy, pretty inexpensive, pretty quick, which can be an absolutely excellent thing. Particularly if you are limited in time or funding or human resources. They do capture data at a specific point in time. Now, this can be a good and a bad thing, right? So maybe you want to collect data at a certain point in time. So when we have things happening in our world politically, or something is introduced, maybe we want to know how people feel about it at that point in time. So we do some kind of attitude survey. But maybe it can also be misleading depending on what's happening in that point in time. So again, use the pandemic. So as we all know, we see these sort of cross-sectional studies all the time telling us, people's attitudes about vaccines, people's level of isolation, loneliness, mental health concerns, for example, right? So what if I happen to collect that data on the same day or right around the same time that a province introduced more restrictions? One might imagine that could influence how people feel, right? About their mental health, about isolation, about vaccines. Oh, I'm tired of it all, right? As opposed to if you collected that data a month later or a month before. So that can be an advantage and a disadvantage, I think. One good thing about the cross-sectional design, though, is that you can collect multiple variables at the time. So it might just be one point in time. But if you're doing some kind of survey or opinion poll, whatever the case may be, you can certainly ask as much as you want, right? So fit in more than one sort of variable. The other thing that you can do is that data can be used in different types of research. So if you wanted to do a randomized control trial and you gave a survey as part of that, okay, great. If you're doing a cohort study, as I said, that Framingham study, those people are filling out questionnaires all the time. Okay, great. So you can use it in different sort of designs, which is good. The challenges to the cross-sectionals, of course, is that, as I said, the data captured at that point in time may not be fully representative because of other things happening at the time. We generally can't determine cause and effect from this type of design. So if that's what you're trying to do, this probably isn't the best type of design, right? And you generally cannot observe trends over time. I think the only time you could do that is if you had cross-sectional data collection embedded within a cohort study that's going over time, right? So just one quick slide on case studies. Again, you'll notice this was kind of pretty low down on the evidence pyramid and the quality. You're simply reporting something you've observed in practice. It's often a healthcare provider writing up these sorts of things. So here's an example, one or more of my depressed patients developed an arrhythmia while taking a particular antidepressant. That might be something novel, might be something new, you might think is interesting and might want to alert other sort of providers in your area about it. You have no comparison group. You often have an N of only one or a very small number. Readers are really left to make their own conclusions. These are generally thought-provoking, for sure, thought-provoking articles, and they're generally meant to be. Often it's because you're seeing something new or different in practice, and certainly might be hypothesis generating, right? So as you learn a bit more about a few of these sorts of patients, that's written up in the case study or whatever the relationship is. Here it's depression and a particular, arrhythmia and a particular antidepressant. But you can perhaps decide or may decide ultimately to move on and study that in a bit of a more robust or rigorous design. So those are the key sort of non-experimental or observational studies. Any questions on those before we sort of finish off with a bit of thinking about experimental design? Just seems like a natural place to say. Any questions before I go there to the RCTs?
I got a lot of questions, but I'm afraid to ask them.
Okay, you get time for one.
No, that's serious. No, I got a lot of comments, but I don't think I'll ask you about it right now. It comes to like the data that you're using for research, patient data.
Mm-hmm.
My observation on that is that the data that's collected on patients is collected by the doctors. Sometimes it's not even told to the patient. So the data you're collecting is not actually, it's subjective data that the doctor is reporting on the patient. Hopefully the patient has a discussion with the doctor, but I've seen in my experience that some medical data that's collected on patients is not accurate.
So
Anyway,
I would just say this. So
I'm saying your data is only as good,
yeah,
as where it is collected.
100%. I fully agree with that. Yeah. And so it's why, in these journal articles and whatnot, it's really so important to pay attention to the methods section that really needs to be robust, well described. And we as readers need to read it carefully as well to understand. But I do want to say this, certainly in a research project, when something is done as part of a research protocol, whatever data is collected really does need to be fully explained to patients and in the informed consent procedure. 100%. That's what's supposed to happen. I want that to happen. I'm chagrined if it's not happening. Really, in a research protocol, I know that there are instances in some evaluation type work, some quality improvement initiatives that are done on the ground in a hospital, in a clinic, in a health authority, where they don't require ethics review. And there may well be data collected there that is not under the stringent way we have to do things in a research protocol. So I will just say, certainly in a research protocol, that absolutely has to be a part of the consent discussion. Just like this here with the trials. If we think about but no, thanks, Asna. It's a very important, it's important, hugely important. We need to be told what's happening with our data, right? For sure. That's another webinar, data usage. So I just want to jump into the clinical trials so we can finish up. And maybe we might still have a few minutes for questions. I'm trying not to go too far on. So here's an example. You have people who have had a stroke that are currently receiving physio, okay? So what if we thought that this transcutaneous stimulation, and those are the little, the little pads you use sometimes in physio, give you little electrical currents, very low voltage for tense muscles. So maybe you think that that will have some impact on, and you can see the second bullet here, walking speed, which is a continuous outcome because we measure it in minutes or in seconds, or the presence or absence of a foot drop, which can be an outcome of stroke, right? And that's a discrete outcome. It's, do I have the foot drop or do I not? Yes, no, as opposed to walking speed, right? A continuous variable. So this might be your example. So we're going to randomly assign some of these patients, all of whom are getting physio, to get this particular TENS therapy or not. Again, here's our famous little graphic. We have our patients over here. So they're already defined. We know who they are. They're being randomly assigned to these different groups. We follow them up and compare the results. Again, whatever our outcome is. Here it's the walking speed, and the foot drop. Now, have you heard of this term before, double blinding? I think you're probably, if you have, probably most familiar with it in terms of a drug trial, where we give, so our treatment group receives the treatment pill, whatever it is, and our control group receives a placebo. So they don't get the new drug, but everyone gets a drug. And double blinding simply means that neither the physician or the manager, whoever's running the study, the researcher, nor the patients, the participants, know who's getting what. That's where that term comes from, double blinding. So no one knows who's getting what. And there are ways to do that in the methodological stage at how we randomize and how we give these pills to people in a trial, whatever the treatment is. Now, did you notice, have you already thought that sometimes we can't blind everything? So in the example I just gave you, for example, about the stroke patients, we cannot blind patients to the fact that they're getting low voltage electrical currents, right? I mean, obviously they're going to know that. Just like a lot of exercise interventions. I mean, if we think yoga is better for back pain than, you know, a particular kind of pill, then obviously people know if they're taking yoga or not, right? So you can't always blind, but certainly in these traditional drug trials, that is the highest level of evidence. Now you might say, well, who cares? Why are we blinding? Well, it's because when you're assessing the outcomes, if you knew who took the great drug, you might be more inclined to actually see the outcome that indicates the drug has worked. So here with the foot drop, for example, if I was observing that as an observer, and I knew, okay, here's a patient coming in who was on the control pill. They didn't take the new pill. I might look at your foot and or measure your walking speed and go, yeah, maybe they were a bit slower than the, so we can introduce our own biases. That's the point of doing the blinding, by the way, as why we do that. So it helps to control some of that, some of our biases. And by the way, there are many. We have many. So here's the major feature of it. We are the ones, investigators are the ones who are controlling who gets what treatment. So it really is under our control, unlike the cohort studies and the case control studies, much lesser extent. Here, we are randomly assigning people to different treatments. So the advantages of it, remember those confounds we talked about earlier, like the coffee drinker with the heart illness and the cigarette smoker and the confound. So because we're randomly assigning people to these different groups, all those different elements of people, our lives, our background, what we call confounders, that could impact the outcome, at least they're partially balanced. It's the flip of a coin, right? Whether someone who is a smoker gets the drug or not. So it's kind of an equal chance, right? So they partially balance. Nothing's ever perfect, by the way, but it partially balances at least. Blinding is possible in a lot of these trials. Not all the time, but in a lot of these trials. And by the way, even when blinding is not possible, as in the case of the stroke patient example, if someone's going to assess that outcome of walking speed or foot drop, blind the outcome assessors. So whoever is going to do that assessment, make sure they don't know which patient got the drug or didn't, or which patient, you know, had the electrical therapy or didn't. Make sense? So you blind the outcome assessors, right? Most stats that we do, most of the statistical tests that we do are actually based on assumptions of random allocation. And so when we randomly assign people, we can theoretically have a better statistical sort of plan. Not that I'm a statistician, but most of the tests we do are based on that. And the RCTs really are the ones that at least allow you some statement about cause and effect, or what is the measurable impact of whatever, exercise A, drug B, etc., which is important. Disadvantages, really super expensive, super expensive. Some trials cost millions of dollars, so quite expensive in time and money. The people who volunteer for the trial may not be representative and often aren't of the population at large. And as we said earlier, right, you can have a potentially effective treatment that is withheld from some patients, and some participants could be subjected to something dangerous. Now, we try to mitigate those things, right? So obviously, clinical trials often have, you know, safety and adverse monitoring boards. Investigators, you know, have a protocol where they will report those things in a certain amount of time, a small window of time, and so on. Trials can and have been stopped when we see any sort of really dangerous impact. So we do have mechanisms in place, but just at the high level, these are some of the disadvantages. In terms of withholding treatment, what we often do with a trial where a patient does not get that wonder drug or whatever it is we're studying, we often do tell them that indeed, if this trial is successful, if you are in the control group and you're getting the placebo, but if the trial is effective, we will then give you the drug. So they generally will allow that to happen when it's possible. I assume that's possible with time and money considerations will be my best guess on that. But so my point is we try to mitigate these as best we can, right? Okay, so let me just make these final thoughts, and then I think we probably still have a couple minutes left for discussion. Okay, good. So here, final thoughts on design. Sorry to rush through all that. As you can imagine, everything I just talked about are full courses, full textbooks on case control studies and cohort studies and trials. So we've just glanced the surface of them, but I hope that gives you some sense of some of the common designs. What I would say is this, that your final design is going to be dependent first and foremost on your research question, right? So if I have a question about prevalence, if I want to know how many people are vaccinated in community A and community B, I don't need to run a control trial to do that, right? For example. So first and foremost, it's going to be dependent on your question. I'm sure that Dr. Twells, when she comes in next week or the week after to talk about research questions, will reiterate this. Think about your questions early. It takes time to go search the literature, find out, well, what's already been done? Can I do this question? Then what kind of design might work best for that question, balanced with this. It's not just about theoretically trials are the best, let's all do a trial. You've also got to balance it on really practical issues. Do you have enough participants? Can you find enough? Can you recruit enough? What kind of time do you have? How much funding do you have? Do you have human resources? Do you need to collect other data? Is it going to be hard to collect that data? So all those things you really do need to think about, I think, to balance them out, not just with the theoretical, here's the best way to do things, but think practically. 3 resources I point out to you all online. This middle one now, I don't know if it's still free anymore. It used to be, but they're all about study designs, okay? Different types of designs if you want to follow up and look at some of the things we've talked about today. And just want to say thanks to Dr. Mugford. So Dr. Jerry Mugford used to use some of these slides. So I stole the diagrams all from Jerry because I don't know how to draw things on PowerPoint. So Jerry lets me use these whenever I teach about research design. So big thanks to him. And there's my email address if you have any questions for me. And happy now, if we've got a few minutes left, Julia, if anyone's still on the lines, I can't see anything here, to chat. Yeah. Go over here. I know Mike had asked me a question earlier about sort of peer review process in research design. Excellent question. It's a huge part of it. So when we apply for funding, for example, at any of the major grant agencies, at least anyway, and even in our own, like even here at Newfoundland Support, we really do think about the research design, and reviewers, particularly at the national level, so CIHR, for example, I think most of the score, I think 50 out of 100 of the points go to the methods, to the research design, and they really pick them apart. They really pick them apart. So the peer review, now you're being reviewed by, you know, anonymous reviewers. They are your peers. They are fellow scientists and investigators. So one assumes, you know, they have the expertise to do that, and I'm sure they do. I didn't mean to imply they don't. I'm sure they do, at least the research design part of it. So it actually fits in a fair bit. When we teach in our graduate programs and have students produce final papers, we tell them all the time, really pay attention to your methods. The fewer questions reviewers have, generally the better. The clearer it is. I know what your intervention is. I know what your outcome is. Oh, okay, I know the participants you're using. Oh, yeah, okay, I see your control group, etc. So yeah, plays a really important part. Yeah. Thanks, guys, for the questions. Yeah, great. Great.
So I know we have about 5 minutes left. I'll give people a chance to think maybe, and while you're doing that, I'll just share. Holly, of course, already mentioned. I'm gonna share my screen now. Holly, is that okay?
Oh yeah, I should stop this. Sorry. Yeah, yes, it's a better view for me anyway. Great.
Just as anyone is kind of thinking about some last minute questions, as Holly mentioned, we do have some sessions coming up. So with our research design series, next week is Dr. Lori Twells. So talking about identifying that research question, why it's, you know, a very important first step. And then a week after is Dr. John Fardy talking about clinical trials. And then in March, we will have our qualitative design sessions, which the registration will be open for those soon. So you can register for all of the events on the NL Support Eventbrite page. We also have a session that's not part of the research design series in March on health economics, and we did that last year, and it was quite a lot of engagement, and it was a good session. So look out for that registration coming up. We do have a training program. This is open to anyone who's interested in patient-oriented research. It consists of these monthly webinars, of course, in-person workshops when we're able to do so. Right now, everything is online. So to receive a letter of completion, you need to attend 6 sessions with at least 1 in-person session if we are holding them within a 2-year period. And of course, you do need to register for each event. If you do have any questions, of course, feel free to reach out to me, and of course, Holly put her contact info there as well, in case you have any questions about today's session or anything patient engagement related. Feel free to reach out to us, and you can sign up for our newsletter as well on our website. Any last questions before we sign off?
I got a question. I know I'm hogging the airtime, but I got a suggestion for a research.
Yep.
For Holly.
Yep.
That would be using the bioelectric impedance. There's a lot of companies out there now that are using this and selling products that will measure a lot of biomarkers just using a weigh scale. I think an interesting study would be to use, get one of these devices and verify it against traditional ways of measuring BMI and other biomarkers in the body.
Interesting.
Anyway, I'm a patient looking for a researcher to do research on this.
So Austin, I was just going to say, so it's useful to know that kind of interest, because part of what we try to do is keep our ears open for, now, do we know anyone with an interest in that kind of stuff? So it's not my area of research, but
No.
we'll have a think about it, but it's not a bad thing to mention at these sorts of events and webinars. So let's have a think about that. Interesting.
Yeah, my point is that as a patient, I would like to say, okay, I got a research idea, and be the originator of the research and recruit the researchers and the people around the research community to engage in the research.
Yeah, it's an example of sort of a patient-initiated research, really, isn't it?
Yeah, there you go.
And that's
That's like taking the pyramid and turning it upside down.
Yeah, it's the latest movement now in patient engagement. The BC support unit, Austin, the BC support unit, go to their webpage. They have a little bit of experience with that, not a lot because it's new, but they did a really good webinar there with, I forget her name, Mike might remember it, Bev someone, I can't remember, sorry, who was talking about the challenges of doing that kind of research, and she has a real patient story as well. Have a look at their website, you'll see it. It's a useful webinar. Yeah. Okay. Anyway, everybody, thanks very much for thanks, Julia, that's great. Thank you for the discussion, everyone. I really appreciate it, and the engagement. Feel free to shoot off follow-up emails.
Thank you.
Thanks, everyone. And again, thank you to Holly for a very informative session that I think will set the stage for our future sessions in our research design series. So everyone have a wonderful rest of your day.
Thanks, everyone. Thanks, Julia.
Thanks a lot.